Yo, what's up, everybody? How's it going? What's up, everybody? How are the experiments going? Anybody had any success yet? Yeah. Sorry about last week. YouTube was down. That was really interesting. So, I've been trying to find time to do a makeup class, but we'll figure it out. Slow. That's no fun. Yeah, I know. I can't tell. So, if we don't have a makeup class, this will be our last class. And I can't tell if the class is a little too difficult. If maybe next time we should make it a little more easy. So, David, the DIY way to quantify DNA, if you look in the file, what you can do is the ladder actually has bands of a certain concentration. So, if you look at our classroom, let me see, Google Classroom. It should be under, let's see, maybe week 3. Yeah, so if you look at week 3, there should be analyzing agarose gel electrophoresis, and it should talk a bit about how you can DIY quantify DNA. So check that out, week 3. And basically what it is just, you know, you look at the ladder, and the bands in the ladder have a certain concentration of DNA. You compare the brightness to your band, and from that you can get a pretty rough estimate, not bad estimate at all, of what the concentration of DNA is. Any other questions? Any questions? Well, what we could probably do today, what I wanted to do is, David, honestly, off the top of my head, I don't know which ladder you got. If you got the 100 BP ladder, it does a similar thing. What you can do is you can just go to the Odin website. Here, I'll just post it in the Google Doc. And it's the same 100 BP ladder. All right. Yeah, so the 100 BP ladder is there. Let me put the image in there. But it should be pretty similar. Ooh, let's decrease that. All right. Any other questions? All right. Let's see if window capture Firefox. All right. So basically, if there's no other questions about your protocols, procedures of how do you get to the end of this class, how you clone your DNA and edit it, I want to talk a little bit about um, if you want to order custom DNA, how you go about doing that, um, all that good stuff. So then, I mean, honestly, nowadays I probably do less and less cloning and more just ordering. It saves you time, and it's really not that much money. Probably for 200. I mean, it's a decent amount of money, but in general, when you're talking about science things, it's not that much money. So for about $200, you can probably what enzymes are gene therapists? Any simple way to separate GFP from the lyses? Um, you could purify the DNA, Daniel, if you're trying to separate the GFP. Oh, separate the GFP protein from the lyses. I don't think so. I don't think there's an easy way, unfortunately. Uh, yeah, I mean, the Biohacking Genetic Design Network is a good place to ask questions. If you join the Odin Bioengineering class, that's a good place to ask questions. The classroom will still be up, so you can ask questions there. Yeah, just ask questions around. All right. So I'm going to bump this up so we can start. All right. So the first thing when I'm trying to do a project is the best place to start is the NCBI. Right? The NCBI, National Center for blah 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 Institute or some bullshit. Biotechnology. Oh gosh. NCBI. No, I don't want to go to BLAST. I just want to go to, I'll just let it load. National Center for Biotechnology Information. There we go. I look here. So this is the basic place where you're going to find all the sequences of any DNA that you want to work with, right? Anything you want to work with, you'll find it here. Generally, what you want to look at is you want to look at proteins. There are times when you want to look at nucleotide sequences and stuff like that, but generally you're going to want to find the protein sequence of the thing that you want. So, for instance, let's see. If we wanted to, I don't know, what is a good thing that we want to do? People are always looking to work with Lux cassette or luciferase. So what we can do is, oh look, luciferase. We can look up luciferase. So luciferase protein, what it does is it creates a reaction, and what that reaction does is it creates light from luciferin. Now we got a lot of luciferase proteins when we look at this. A lot of them are pretty huge. So the first thing we need to do is figure out, well, what type of luciferase do we want, right? There's so many of these. So let's just do a web search. Best luciferase. Let's see what people say. What's the best luciferase? All right. Okay, so here. So the classes are based on interaction. Luciferase that are most widely used are beta luciferases, vanilla luciferase, and a modified deep sea shrimp luciferase. All right. So where's this figure 8.1? All right. So what we want to find is Photinus pyralis. Seems to be the most commonly used luciferase. All right. So let's see if we can just search for it. Photinus. No? Nothing? You're not going to give me... Oh, I can't press enter. I have to click search. Ugh, I hate when it does this. Okay. So let's say protein name luciferase organism Photinus, right? Is that what it is? Photinus, not tennis. Photinus. All right, now let's search. Yeah, there we go. All right. So we got a bunch of different luciferases. Here's the Photinus pyralis one. One thing to watch out for is not all these sequences are full sequences, right? So a lot of times what you'll see is this stuff right here where it says like partial. Now that literally means it's just a partial sequence of the protein. So somebody did some sequencing or identification, just did partial and uploaded it to the database. Not what we're looking for. What we're looking for is a full sequence, right? So this one does not say partial. It's much bigger. It looks like a full sequence. Let's click up. And so now this gives us all the information about it. Organism, gene, different parts of the protein, and all this stuff. And this is the sequence we have, right? So this is the luciferase gene. Now what we're trying to do, let's say, is we're trying to make ourselves glow in the dark, right? Or just glow. So we want to make ourselves glow with luciferase. So we're trying to insert luciferase gene into mammalian cells, right? So we got the sequence here. So the next thing we need is we need a place to order it. Now there are many different places to order the gene, but there are a lot of things to take into account, right? So the first thing to take into account is what parts of the gene do we need, right? And so let me open up Snap Gene. Let me see if I can pull that up. All right, let's open up a file using Snap Gene. Find some plasmids. Where are the plasmids at? Alright. Let's find a human plasmid. Alright, let's open it up. Let me bring it up. Window capture. Alright. So this is a snap gene. So you should all have downloaded snap gene by now. I'm just pulling up a mammalian sequence that I know. What you can do is you can, you know, find some mammalian plasmids on add gene or download from snap gene or whatever. All right, so let's look at the map. So this is the structure of our plasmid. Remember, it's got these different things. It's got antibiotic resistance and all these other things. So in this plasmid, our gene of interest is this IGF-1 gene, right? So this is the gene we use for our frog genetic engineering kit at the Odin. And the IGF-1 gene, what it does is the insulin-like growth factor 1. It increases growth factor production in the body and makes organisms bigger because of that, right? So if we were to create our own plasmid, what we'd want to do is we would want to take this IGF-1 gene, remove it, and put in our gene, luciferase, right? That's what we're trying to do, is put in luciferase. Now, we can't just use the luciferase sequence by itself, remember, because what we need is we need promoters, like the CMV promoter, and we need terminators, like this BGH polyA signal, and that's what we need in order to really express this gene. Okay? So if we were to synthesize this, it would be not just our gene, but it would also need the CMV promoter and enhancer. So what we can do, let's just copy all the way up to the starting methionine. Edit, copy top strand, open up a notepad file, paste this sequence in there. And this is CMV. All right. And now we need the terminator portion. So let's just go from the stop codon, TGA, copy all this poly-A signal, copy top strand. All right, so now we have the promoter and terminator. But if we go back to our sequence, what we have is we have the amino acid sequence, and we need a DNA sequence, right? So let's copy this. Let's find out what we do with that. Now, generally, when you have a sequence, amino acid sequence, or even a DNA sequence, and you're moving it between one organism and another, you want to do this thing that's called a codon optimization. Now, a codon optimization is just a so they've kind of found out that in DNA, right? So three DNA bases code for each amino acid. Those three bases that code for amino acids are called codons, right? They found that in organisms, the codon usage, because there's more than one codon for each amino acid, right? The codon usage between species is different. So bacteria have a different probability of using each codon than humans do. And this is related to a lot of different things. Now, they haven't really shown that this is super important for gene expression, but everybody does codon optimization because why not? Nobody's shown that it actually hurts gene expression. So there are a number of websites that do codon optimization. Um, the most popular one is probably IDT, right? So let's do IDT codon optimization. Let's see. Is IDT not codon optimization? All right, let's try this one. God darn it, where's the tool? Why don't you just let me use it? Why does nobody just like... No, same thing. Come on. GenScript, man. Freaking scamming me. All right, see what happens. If I click on try it now. Oh my gosh. Nobody wants to make these available for free. FYPH. All right. So here we go. Copy and paste. So luciferase, the original sequence. All right. So we go to here. Let's copy. Please don't freak out because I have all those numbers in there. Oh my gosh. Freaking out. All right. Why it's gotta be so difficult? I gotta go through. Wait, actually, I might be able to do it in here. Let's see. Let's change the formats to FASTA text. All right, there we go. Copy. Let's try this again. Clear table. All right. Luciferase. All right. Oh gosh, come on. Why isn't it taking the whole sequence? Clear table. This is annoying. All right. I'm going to paste it in Notepad and try to remove those end of lines. All right. Let's try it again. Luciferase. All right. Oh my gosh, I got it. All right, so the organism we're trying to get it is human. All right, now let's click optimize. Man, should not take this long. So basically, it's trying to take this amino acid sequence and find which codons are most widely used in humans for each amino acid, and then give us the nucleotide sequence. It shouldn't take that long, but it seems like it is. I mean, come on. Take like 5 seconds. I could probably do it by hand faster than this. Uh, all right. Well, while we wait for this stupid thing to finish. So, one place that is great for ordering DNA is GenScript. GenScript does large scale or large synthesis, and they do a lot of different stuff, unlike some other companies like GeneWiz and places like that. They also work with pretty much anybody, so check them out. It doesn't matter where in the world you are or what's going on, check them out. They're pretty good. Now, if you're trying to do work with uh, creating your own, you probably want to choose gene synthesis. So there's a couple different things that they have. They have DNA fragments. They have, you can generate oligo. Oligo is just like a small piece of DNA. DNA fragment is generally a larger piece of DNA. And gene synthesis is when they take a gene and put it into a plasmid. Now this is probably always what you want, right? Because you don't want to synthesize just a gene, because that just gives you this piece of DNA that you then have to put into a plasmid if you want to replicate it and use it. So it's usually best that you order the DNA and have it inserted into a gene, right? And here's kind of gives you an idea of what is available, the turnaround time, and how much it's going to cost, right? So generally, what you're talking about, so 23 cents a base pair if you are going for the cheapest. It'll take 17 days though, right? Um, if you want to pay 35 cents a base pair, then it'll be, 10 days. So let's see. So if we have right now, is this finished? It didn't finish. What's going on with this gene thing? So if we have from our luciferase 550 base pairs times 3, because there's 550 amino acids, 3 base pairs per amino acid, it's 1,650 base pairs. So if we were to do it at 23 cents, that'd be about 380 bucks. Just to stick the gene without the promoter or terminator or anything in there. Right? At 35 cents, so if we were to do 35 cents, it's about 580 bucks. So savings about 200 bucks if you're willing to wait an extra week or so. So cloning into a plasmid costs an extra $49. So let's see what we got. So it'll probably take, well, about 20 business days. But hey, we're saving a bunch of money. It's great to do, easy to do. All right. So let's see. What we want to do is order something, right? So let's go to get a free quote. GenScript does this weird thing where sometimes they just let you get the price, but other times they like mail you a quote or something like that. So yeah. And so for this, let's do luciferase. So when it says 5' sequence here, search enzyme, this is the enzyme that generally you're doing restriction digesting with. So if in the future you want to add or remove or put in stuff, this is where you get to choose what enzyme you do. So we don't really care about that too much unless you got some stuff to go with. So, all right, what we got is we got our protein sequence. Let's paste it in here. And we don't really care about the restriction sites. So let's just go here. Let's optimize for human. Add an additional stop codon. We have a stop codon. No, we don't have a stop codon. So yes, we want to add an additional stop codon. So usually stop codon is represented by an asterisk or a star. All right. So now they have the cloning, and you get to choose the vector. Vector arc cloning. Let's see what vectors they have. How come it's not letting me choose? Oh, these are my archive vectors, or my share vectors. Don't they have their own vectors that they could put it in? Is this all we really have is the PUC 57 and stuff like that? All right. Maybe we can't do this with the economy synthesis, which sucks. Let's try just normal gene synthesis. What we're trying to do is get it inserted in a custom plasmid. Oh yeah, here it is. Crap. All right. So we can't use economy synthesis if we want to make a human gene. So what we do is put in our sequence, codon optimized for human, add additional stop codon, yes, standard service. All right, so here when we have the cloning, right, this is which vector they're putting in. Now, certain vectors already are can express genes, like pcDNA 3.1. So if we want to look up what the pcDNA 3.1 is, we can probably just pull it up in SnapGene. So we can go to File, we could go to Import from, let's go SnapGene, search. pcDNA. All right, it's pcDNA 3.1 plus. There we go. So here is the sequence of the plasmid that we are working with, right? So, oh gosh, is it? Yes, All right. pcDNA 3.1. So this plasmid already has CMV enhancer promoter and already has the BGH polyase signal terminator, right? So this plasmid already has everything we need in it. For the low price of 35 cents a base pair. So we know this will already have the promoter, it'll already have the terminator, everything will be ready to go. Now, if we say also want to use this for some gene therapy or genetic engineering, what we want is we want a custom plasmid preparation. Now, how much plasmid is used in gene therapy? That's a good question. Now, we had a couple papers on it that we read about, but depending on the organism, if you're talking about a small organism like a frog or a mouse, you're probably going to use less than a milligram for a gene therapy experiment. On the order of 50 to 100, couple hundred micrograms, right? But for a human, depending if you're trying to engineer your whole body, trying to engineer like all your whole body, systemic luciferase, you're probably going to need a ridiculous amount, maybe on the order of, 2 grams, 2,000 milligrams. But let's just say you're trying to inject it in your arm muscle or put it on the skin on your arm and make that glow from luciferase. So we probably need somewhere around 10 to 20 milligrams. Let's go with 10 milligrams. No other requirements. And so the buffer we probably want is just water or PBS. I mean, you could do PBS also. All right. So here we go. We got the sequence. We got all this stuff. Let's say luciferase. And let's add it to our cart. And click submit. Nothing happened. Why is nothing happening? This field is required. Oh, we need to choose the cloning sites. Which enzymes do we want to use? So let's go and look at our enzymes. What do we have in the multiple cloning site? We have NHE1, ND3, BAMH1. We could choose basically any of these. So let's just say we want to choose BAMH1 and XHO1. All right. BAMH1, XHO1. Oh. So, all right. So let's see what price they're going to give us. Quote. All right. This is something else. Let's delete that. All right. So our gene synthesis, we need to get a quote for that. It says, if we produce enough plasmid DNA, This is going to be about, so that's like 700, and what we calculate. So 35 cents a base pair. So we're looking at somewhere around $1,300 to make the luciferase gene therapy. Now understand, this isn't really realistic. Luciferase requires luciferin, so you're not really going to make yourself glow if you spend $1,300 and inject yourself with luciferase, 10 milligrams of plasmid DNA. But you could possibly do other things, right? It's not unreasonable to think that by injecting 10 milligrams of some gene therapy into your muscle, it would change it, right? All for the low price of about $1,300. Now, the thing is that uh, is that what if you wanted to, if we what we wanted to do was um, we wanted to, let's say, make a gene therapy from a gene that is already available in humans. So for instance, a gene like VEGF. So VEGF is vascular endothelial growth factor. Now what it does is it stimulates angiogenesis, or the formation of blood vessels. Now if you were an athlete or something like that, having extra blood vessels in your muscles is a good thing, right? It increases the oxygenation and blood flow to your muscles. Now, with most genes, there's probably a number of different types, right? So the great thing is that GeneWiz, what they have is they have a service called ORF-cDNA clones. Now that means open reading frame, which is just means a gene in some weird nomenclature. A human gene that you can find it already in a plasmid, right? So let's find a gene. So we want to find VEGF. Let's search for VEGF. All right, so we want human VEGF, not Norway rat or African clawed frog. All right, now there's all these different forms of it, right? VEGF-C, VEGF-D, VEGF-C, platelet-derived growth factor. All right, so now our first goal is to find out, all right, which VEGF do we actually want? So let's start off with the Wikipedia page, right? Let's look at the different isoforms. All right. Oh, geez. All right, let's try something else. This looks like it doesn't really say. It just has a bunch of different listings. So what we can do is we can see if anybody's ever done a gene therapy with VEGF before. All right, there are some people. So let's look up and see what isoform of VEGF they have used. Blah, 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 blah. VEGF A165 is the first one they measure. All right, here we go. Gene therapy clinical trials with VEGF family members. Okay, looks like almost everything is with VEGF A165. Okay, so let's look for that. So we are looking for 165 amino acid VEGF A. VEGF B. Let's just search for VEGF A. VEGF A. See what we get. All right, VEGF 165. Here we go. All right, this is the actual protein, it looks like, and not the gene. We want the gene. All right, maybe they just call it VEGF 165. Here. Let's check ORF clones. I got 8. Let's see. All right. So we got a number of things. NRP, VEGF A, right there. All right, let's check out this. VEGF A. I think this is the same one, but let's check it out. All right, here we go. So let's look at the sequence for this. So that says this one is 576 base pairs. Um, 576 divided by 3 is 192, so that's not the one we want. Let's try the next one. That's 1,000. That's too much. Let's see, can I just scroll down? Yeah, here we go. Let's just look on this page. 165. All right, we got one match. That's 165B. Uh, 65 times 3 equals 495. So let's try looking for 495. No, there's no 495. Do they not have this isoform of VEGF that we want? What's the deal? So they have 444 divided by 3 equals 148. That's too small. 525, 630, 648, 1,000, 576, 492. 414. Okay, maybe they don't have this one, which doesn't make any sense. Why would they not have it? VEGF 165. Looks like 121 has also been used. So that's possible. 121, what is that? 363? I didn't see that. I didn't see anything that was 363 in size. Oh, disappointing. So they don't have any of the VEGFs that we want, unfortunately. Yeah, I mean, theoretically, we could try one of the other ones, but that's probably not the best decision when doing a gene therapy because you have no idea if it'll have anything like the same effect, right? So you'd want to try to stick to the ones that are available. Labs. Hey, Pamela, it's not possible to do the labs in class, right? So all the labs we have left for this class involve skills that were learned in the first half of the class, right? So purifying DNA. Once you purify the DNA, you need to do restriction digest, which is just basically using a pipette and running things out on gels, right? Which you should have learned in the first half of class, which we, demonstrated. And then doing a transformation, which maybe is the only skill we haven't demonstrated. We do have plenty of protocols with pictures that are available to show you how to do that. So everything for the second half of the class was basically reusing all the skills we learned in the first half of the class. What I'm trying to do now is give you more skills. So if you get everything working, what you can do is you can actually use the skills that you've learned in this class to do something completely new and different. Anyway, so our VEGF thing failed, but you get the idea, right? If we want to find a gene, a human gene, that we want to overexpress in humans, right? We can probably find it at GenScript. If we want to order a new gene that's not in humans, what we can do is we can have GenScript do the gene synthesis, right? Put it into a plasmid and produce a lot of that plasmid for us that could be used for a genetic engineering or gene therapy experiment. You can do the same thing if you're doing it just for bacteria or yeast or anything like that, right? Generally for bacteria or yeast, you're going to use a lot less. So it's going to cost you a lot less, right? It's probably going to cost you half. So you wouldn't have this $679 charge. What you're going to have is you're going to have just the cost of the gene synthesis and cloning, right? They send you 4 micrograms, which you can then transform and purify yourself. So you have plenty of it. So, you're talking about $600 for completely new gene to be made and put in a plasmid. That's quite expensive. But if you're synthesizing something new, you're always going to have that cost of gene synthesis. So even if you wanted to not pay the $49 for cloning, you're still generally going to have to pay between $200 and $750 to have your gene synthesized. And in case so you don't mess it up, that $49, it really isn't that much to do it all. Now, it it totally is expensive to do this, right? But designing your own uh, genes or DNA, right? You're talking about you can alter the DNA of a living organism. Perhaps sell it to your friends, talk to me, maybe we could list it on the Odin and sell it if it's something interesting. Hopefully you can make your money back. There's also a lot of websites that you can use to try to raise money for. One of them is experiment.com. Experiment.com is kind of like a science crowdfunding website where you can propose science projects and try to raise money to complete the science projects, right? It's pretty nice. And, I'm sure if you wanted to raise $1,500, it probably wouldn't be all that crazy. Probably find enough people to pitch in to get you that $1,500. So there are a lot of ways to get the money if you can't afford it, right? Pamela, making plates. Making plates is generally considered a basic lab skill. Um, we have instructions, we have videos that we've posted about how to make plates. Um, they should be available. If you can't find those, let us know and we can make those available for you. Uh, One of the reasons we called this class an advanced class is because we expected people to have the skills to be able to make plates and transform bacteria, which is taught in our 101 class. Um, yeah, this class is definitely more difficult. What we wanted to do in this class was go over more advanced skills that you could learn and use, and hopefully you've gained that. All right. So, does anybody have any questions about stuff we covered today? Um, Stuff going on in the class? If you have a specific question about stuff in the class uh, that I can help you with, something that you're having difficulty with, that you're struggling with, is there anything before we sign off for today? Anything? Can I cut gel? Oh, of course. Are you talking about your agarose gel? If you're running your agarose gel in a small container, yeah, you could cut it to any size you want. It shouldn't be any problem. It shouldn't be any problem. Um, yeah, right. So the gel, you could cut the gel in every single way. You don't want to really reduce the size, the length of the gel because you want the gel to be able to the DNA to move a certain distance. Otherwise, it doesn't really separate out well. So you want to keep the length, within, you don't want to go below half the size of the gel molds. Good night, Matt. Hope all's well. Um, Once you start going smaller than that, you're gonna have real difficulty separating the DNA. But like width-wise, the number of wells and everything like that, you can cut that down to as small as you want. Yeah, it's possible. Are you trying to get a faster? Uh, Pamela, if you have any questions, specific questions, just let us know what specific questions you have about the instructions, and we'll try to help you. Um, David, about transformations, there's so many different steps and things that people do. We have not seen any increase in efficiency if you ever put it on ice. You can do whatever you think is good or not good, with your transformations. But, there's some stuff that's just like voodoo more than it actually helps, right? So generally, people don't know the exact reason why a heat shock works, but generally, people think it's because of convection, right? So as you change the temperature um, from hot to cold, what it does is it moves the DNA around more and it sucks it in different directions, right? So DNA will move from areas of cold to areas of hot. And when that happens, it should help the DNA move inside the cell. The temperature increase should also help disrupt the membrane of the cell a little bit, because it's a lipid, and when you heat up lipids become more, less solid. They become more, malleable. So putting it on ice again after you heat shock, the exact reason I wouldn't really, I mean, maybe to try to move the DNA around even more, you know, cold, hot, cold, hoping the DNA moves a bunch. But uh, honestly, in my experience, I have never really done that step. And we've tested it out in the lab, and we've always had reasonable efficiency. But if you're having trouble with the transformation, what I would do is, if you're having trouble with the transformation, just transform the regular plasmid. Don't transform your digestion and ligation. Just transform the regular plasmid and make sure you can get colonies with that. If you can get colonies with just the regular plasmid, then move on to the digestion ligation transformation. If you can't get colonies with the regular plasmid, well, then you need to figure out what's wrong. A lot of times the problem is you're not outgrowing for long enough. So after you do the transformation, you're not letting it grow in the LB or SOC or whatever you're using. You're not letting it grow in the media for long enough so it can replicate the plasmid. The other thing is you might have too much bacteria in your tube. You might have not enough DNA that you're adding to your tube. You might heat shock for too long. So remember, don't heat shock for too long. If you go over about 20 or 30 seconds, what you're doing is you're going to kill a bunch of the bacteria, right? So 42C is pretty warm, and you don't want to kill all those bacteria. So try to keep it pretty close to 42C or lower. Definitely don't go above that. But yes, get the transformation to work with plasmids like the GFP plasmid or the P-Dusk or P-Dawn plasmid first. Then, if you have that working, then I would say move on to the ligation. Um, but your first step should just be get the transformation working. Raul, I don't know of any viral vectors that are activated with antibiotics. So I don't really know. Um, I can't answer your question. Sorry. Partial cell reprogramming. You're talking about anti-aging stuff? Uh, um, you know, I, this stuff doesn't really say much on the websites I'm going to about gene therapy or viruses or antibiotics. So, I'm sorry, but I really don't know. Um, but yeah. Hey, Raul, I'm sorry, I don't know, Raul. All right, I'm gonna take off, and if you have specific questions, just let us know. Uh, and I will try to answer your questions on the Google Classroom. If not, well, I will talk to you soon then.